Right, and now we can move to the next topic, which is how things are moved across the plasma membrane. So, as you know, the plasma membrane, being a bilayer phospholipid, will not allow anything non-hydrophobic across it. So, if something is hydrophobic, so it doesn't have any form of electrical charge, not even partial, it will go through the plasma membrane quite effectively and um, quickly. And also polar molecules can go through it if they are of small size, with the limit represented by glucose, which does not cross the plasma membrane. So polar molecules can cross the plasma membrane to a point, to a size, and a good reference for a limit is glucose. Anything above glucose cannot pass, and also glucose cannot pass. Water can pass. It's a polar molecule and it can freely diffuse across the plasma membrane. Ions cannot pass because ions, such as chloride, sodium, calcium, potassium, or anything electrically charged, cannot pass because they are electrically charged. So, to allow big molecules and charged molecules or big charged molecules to go across the plasma membrane, we do have transport proteins. So we can have just passive channel proteins, such as aquaporins, which facilitate the passage of water across the plasma membrane. So we know that water can go through the plasma membrane, but through the aquaporins can go much faster. And also we do have other transport proteins, such as carrier proteins or channel proteins. It is very important that you remember that transport proteins are specific. Okay, they don't allow a specific movement of chemical species. They only allow the movement of one chemical species or a subgroup of chemical species. So we can classify, because we scientists like to classify things to bring order into chaos. The different kinds of transports across the plasma membrane, depending on whether or not we need a carrier or whether or not we need free energy. So first of all, we have passive transport, which is the transport across the plasma membrane, which is driven by the gradient across the plasma membrane, which can be a chemical gradient for a non-charged molecule or an electrochemical gradient for a charged molecule. We can have simple diffusion, as we said, of hydrophobic substances or polar substances that are smaller than glucose across the plasma membrane. We can have facilitated diffusion through channel proteins of a specific chemical species across the channel. Or we can have a carrier protein, which is not a channel. It's a protein which changes its conformation to allow a substance to go from one side to another side of the plasma membrane according to its gradient, such as the glucose transporter, for example. And then we do have active transport, such as the sodium-potassium pump, which transports species against their chemical or electrochemical gradient through the hydrolysis of ATP, which, as you know, is the energy coin of the cell. So we should remember that the sodium-potassium pump is a very important protein. Almost a third of the energy of a cell, and that can get up to 75% of the energy reservoir of the cell, for example, in neurons, can go into feeding the needs of the sodium-potassium pump. And this is due to the fact that the cell needs to keep the plasma membrane potential and gradient of sodium and potassium constant. So the resting membrane potential is actually not resting at all, because if it's true that in resting conditions the plasma membrane is not very permeable to sodium, while it is permeable to potassium, but there's not a strong electrochemical gradient that pushes potassium outside of the cell. Notwithstanding that, you always have this leak of very slow but constant leak of sodium inside of the cell, and this very slow but constant leak of potassium outside of the cell. And that's, pretty much like a slowly sinking boat, yeah? The sodium potassium pump, what it does is to spend a lot of energy to pump potassium inside of the cell, 2 ions of potassium inside of the cell and 3 ions of sodium outside of the cell per molecule of hydrolyzed ATP. It's basically like having a ship that floats by having holes in its hull, but there's a pump that constantly pumps water outside. That seems a stupid way to keep afloat, but it's actually very smart because it allows the plasma membrane potential to react very quickly to perturbances in the external environment. This is particularly important because the plasma membrane potential is one of the main ways the cell interacts with the environment and how it transmits information across the different compartments of the cell. I am an electrophysiologist, so I devoted my life in studying and characterizing the plasma membrane potential and their changes in health and disease in neurons. So I am obviously a little bit biased, but yes, sodium potassium pump, very important. So inside of the cell, we do have all sorts of substances which are trapped inside by the plasma membrane, such as proteins, sugars, amino acids, nucleotides, and ions. And so for this reason, water will always move inside of the cell for osmosis. And if this is unregulated, the cell will burst. The sodium potassium pump also plays a key role in keeping the osmotic balance between the inside and the outside of the cell. So what the sodium potassium pump does is to actually keep things regular. So for the same reason, I will not show you this animation of the sodium potassium pump in the video because otherwise my poor computer will crash. But I will upload the PowerPoint presentation on Blackboard, so you will be able to see it there. So another mean of transportation across the plasma membrane is the receptor-mediated endocytosis, which is carried out thanks to these clathrin-coated pits, where a membrane protein recognizes the ligand in the extracellular space. Once the ligand binds to the receptor, the clathrin-coated pit will invaginate inside of the cytoplasm of the cell, form these caveolin and clathrin coated pits, forming the vesicle. The vesicle will lose the clathrin coat, and then the receptors can be recycled onto the plasma membrane, and the endosome can be converted into a lysosome, and the ligands can either be recycled or destroyed. So the receptor-mediated endocytosis is a very important and highly regulated mechanism. It's involved in many different cell functions, including immune function, the reuptake of neurotransmitter, and the communication between different cells. There are different kinds of coating materials. One of them is clathrin, which works by forming these bee nest hexagon structures around the vesicle, and also COP1 and COP2. Of course, like each one of these proteins is involved in different steps of the transport, and each one concentrates different proteins for internalization. So here you can see a beautiful EM series of images of clathrin labeled with LDL ferritin, which is electron dense, and then which allows us to see the invagination, which brings to the formation of the endosome and the disassembly of it once the endosome is formed. So endocytosis works in a similar way to endocytosis can exist in different forms depending on the size of the molecules or the corpuscle that we're talking about. So we can have phagocytosis, where rather than having coated pits, we have pseudopodia, which are emitted by the cell as processes to envelop a food or other particles such as bacteria or other larger things present in the environment surrounding the cell. Or we can have very small pits, allowing the internalization, the slow internalization of small molecules. And then, of course, we can have receptor-mediated endocytosis, which uses coated pits and specific receptors. As much as we do have endocytosis, we do have exocytosis, which is a similar mechanism, which allows on the other hand, to take a vesicle from inside of the cell, fuse that vesicle with the plasma membrane and release the content of the vesicle in the extracellular fluid. This is the main mechanism through which neurotransmitters are released within the central nervous system, and synaptic transmission is the way neurons communicate with each other, and synaptic transmission and synaptic plasticity are the ways our mind works, our memories are encoded and stored. So knowing this is very important also to know yourself in a way, as Socrates always reminds us. And of course, this process doesn't occur in uncontrolled way. You will know more about the snap and snare mechanism, for example, through which the vesicles fuse with the presynaptic membrane, or with the plasma membrane of the cell, leading to the release of solid into the extracellular space. But I will not enter into that level of detail because you will be exposed to it in more specific lectures. So when we talk about exocytosis and endocytosis, we truly are talking about processes which constantly occur, and which constantly occur also within the system of endomembranes, including the Golgi apparatus, the trans-Golgi network, and the endoplasmic reticulum. And I think it's kind of important nowadays to know that these mechanisms, which are so important for the communication of the cell between cells and the interaction with the environment and whatnot, can be hijacked by pathogens such as viruses, which will hijack not only the endocytosis and exocytosis mechanism, but all the mechanisms of vesiculation and transport across the system of endomembrane. And one clear example is HIV or the classic influenza virus.